The next sort of transport is cell-mediated transport, and this is the movement of ions and organic substances in substrates. So characteristically, characteristics mean that it is a specific protein and one set of substrates. There is a limit on the saturation, and so if saturation is reached, then there will be no movement, and there is also this movement is also regulated by hormones. You can have carrier-mediated transport, where you can have co-transport, where two substances move in the same direction at the same time, and then you can have counter-transport, where one substance will move in while another one moves out. And then you can have facilitated diffusion, which can be passive, so where carrier proteins transport molecules that are too large to fit through channel proteins, such as glucose and amino acids. And these molecules bind to the receptor sites on the carrier proteins and help move through into the cell. So an example of this is the hormone insulin, which needs to be attached to the glucose, which then needs to attach to the carrier mediated on the cell in order for the actual cells to be able to accept glucose. No insulin, no glucose moving into the cells. So here we have the extracellular membrane, so a diagram showing you we've got the cytoplasm, we've got the glucose molecule, and we've got it linking up with the carrier protein, and then it moving. We've got gates opening and gates closing, and glucose is released into the cytoplasm. We can have primary and secondary active transport. So active transport moves substrates against the concentration gradient, and because it's moving it actively, remember that it requires energy such as ATP. And then you can have ion pumps. So ions are forms of electrolytes, which means they are charged molecules. And an ion pump is an example is the sodium pump, the potassium pump, the calcium pump, and the magnesium pump. And these are exchange pumps, which can then you've also got exchange pumps which can counter transport two ions in the same direction. If we look at passive and active transport, you can have the sodium-potassium exchange pump. This is where active transport and carrier-mediated transport, so we've got sodium ions, which is Na+, where they move out, and potassium ion, K+, moves in. It takes one amount of energy of 1 ATP to move 3 sodium ions and 2 potassium ions, where they exchange for each other. So here is the sodium-potassium exchange pump, which we'll talk more about in this topic, but also in the second semester topic 1005. So again, if we look at the diagram, we've got the extracellular environment, and here we've got 3 purple round balls representing sodium molecules, which are sodium ions that have got a positive charge. And then on the inside, we've got smaller potassium. They're the sort of the little red balls, and they're moving into the cell, whereas the sodium is moving out of the cell. And for that to occur, we want sodium to move out of the cell and we want potassium to move into the cell. It requires energy, and it requires ATP and that is transformed into ADP, adenosine diphosphate. And that adenosine diphosphate, like we showed you in the first lecture, becomes adenosine triphosphate in the process called a cellular respiration, where you have glucose in the presence of oxygen that transforms adenosine diphosphate into adenosine triphosphate, ready to be used for energy. Cell-mediated transport, you've got secondary active transport, where the sodium concentration gradient drives glucose transport, and ATP energy pumps the sodium back out. So here we have glucose molecules and sodium. So we have sodium moving in with glucose coming into the cell, but the cell doesn't want to have too much sodium. So it would rather be moving the potassium into the cell and moving the sodium out of the cell. So it achieves this by the sodium-potassium pump and using energy. So you can see in the far left diagram where glucose and sodium are moving into the cell, and then that is released in the next diagram. But next to that protein that has allowed that movement to move through, you've got the sodium-potassium pump, where potassium is moving into the cell. 2 potassium ions are moving into the cell, while with energy you've got 3 sodium ions moving out of the cell with the help of adenosine triphosphate, which is transformed into adenosine diphosphate. The other form of transport is called bulk transport, and this is where the organelles within the cell actually form vesicles to allow movement of materials out of the cell or into the cell. So you've got endocytosis, which is within the cell, and it requires energy, so it is receptor mediated, and then you've got pinocytosis and phagocytosis. So we'll look at those now. So endocytosis inside the cell, receptor-mediated endocytosis, where you've got glycoproteins binding to target molecules called ligands, and that these coated vesicles carry the ligands and receptors into the cell. Here's a diagram of it. We'll go through it slowly, starting at number 1. So again, if we just look at the orientation of the diagram, the purple part of the diagram is the extracellular fluid. The line with the little blue bits with the little sort of arms hanging on it is the actual cell membrane, and the buff color underneath it is the cytoplasm inside the cell. So this is a diagram showing you receptor-mediated endocytosis. So if we start at number 1, we've got the ligands, and they bind to the receptors in the plasma membrane. So you can see there, they've got the little arms up, and they've got the receptors binding to them. What happens is we move to number 2. The areas are coated with ligands form deep pockets within the plasma membrane, and then those pockets pinch off to form the little circle, which you see as number 3. And then it becomes known as a coated vesicle. That moves down to number 4, where it is fused with lysosomes to form a secondary lysosome. So we've got the primary lysosome is attracted to the coated vesicle and then joins onto it, and then we move to number 5, which you can see is again a circle, but it's part secondary lysosome and part ligands, and you can see that the ligands are now coming off and being absorbed into the cytoplasm. Now we've finished that part. We've got detachment, and we've got the lysosomal and endosomal membrane separating, and then we've got it moving back up to fuse again with the plasma membrane, and then it will be free again to bind with the ligands. And this is happening all of the time in all of our cells. So endocytosis is also known as a process called as pinocytosis, which is cell drinking, and phagocytosis. So phago, where you have a pseudopoda, a false foot, and it goes out and it engulfs things like bacteria and dead cell particles. So exocytosis outside of the cell. Granules or droplets are released from the cell into the external environment. So here we have a highly magnified 20,000 times of magnification, but what they've been able to capture and colour for you is this example of pinocytosis. So here again, if we look at the picture, we've got the bloodstream up top, so that is the blood vessel moving past, and then you've got the cytoplasm, and then you've got the surrounding tissues. And here you can see that the little vesicles are being formed, and they're moving in through the cytoplasm, and then they can move through to the bloodstream where they can be released. Here's an example of phagocytosis. So we have the phagosome being formed around the bacteria. So bacteria tries to enter the cell. It's formed, the cell pushes its membrane around it, and it enters the cell in a phagosome. Their lysosomes then join onto the phagosome, and it fuses with the phagosome, and it destroys the bacteria, and then it moves to the other side of the cell membrane where it is released by exocytosis. So we remove the waste products from the cell. Here's a table from your textbook. I'm not going to go through all of them, but you can see that there are many different ways that many different mechanisms that are involved in moving fluids and particles and ions and nutrients and waste products across the plasma membrane. Some is passive, some is cell-mediated, some is requiring energy. So that's the end of lecture 1. I suggest now that you can re-watch this part of the lecture or go and have a look through your notes. And when you're ready, I'll join you for lecture 2. Bye for now.